This is the Turf Zone Podcast, your central information and news hub, bringing together professionals from turf associations across multiple states to share things to help you and your business. Brought to you in partnership with our friends at the Mississippi Turfgrass Association. Now, let's get in the zone.
Welcome to the Turf Zone. In this episode, we feature an article titled Genomics-Enabled Breeding for Turfgrass, written by Hongxu Dong. Turfgrass breeders seek to improve grasses for a variety of end uses. The ultimate goal is a variety capable of withstanding challenging environments without unnecessary inputs and pampering, all while providing an aesthetically pleasing appearance. Through plant genomics, we can sequence the genes and then study the relationships of those genes on typical characteristics. Plant genomics aims to characterize the genetic compositions, variations, structures, functions, and networks in plant genomes. Through it, we will gain better understanding of genetic mechanisms of traits of interest in order to produce better turfgrass varieties. A superior grass growing on the plains of Africa may have value only to the cattle or wild herbivores that graze it. Although it has intrinsic value, its economic value is limited. If this plant is collected, evaluated, used in a breeding program, production tested, and released as a cultivar that is planted on a large acreage, its value is greatly improved. This is the basic procedure of Bermuda grass cultivar development. Bermuda grass is now the dominant warm-season turfgrass species in the southern U.S. and has contributed billions of dollars to the economy. Bermuda grass is a warm-season, fine-textured turfgrass adapted to a wide range of environmental conditions. It is a drought-tolerant, durable, and versatile turfgrass that can be used in many settings, including golf courses, athletic fields, home lawns, and as utility turfgrass. Common Bermuda grass and African Bermuda grass are the two primary species used in breeding improved Bermuda grass. Bermuda grass species are largely self-incompatible, meaning that they rely upon cross-pollination rather than self-pollination. African Bermuda grass is a diploid, meaning it has 2 copies of chromosomes. It readily crosses with tetraploid, having 4 copies, or hexaploid, having 6 copies, common Bermuda grass. Hybrid Bermuda grass has been successfully employed in the development of turf cultivars. In fact, most commercial turf-type Bermuda grasses are crosses of African Bermuda grass and common Bermuda grass parent plants. Interspecific hybrids such as Tifgreen 328 and Tifway 419 are well-known standards for golf courses in warm, subtropical, and tropical climates. A plant germplasm is a collection of living materials such as seeds or plants that are maintained for breeding, preservation, and other research uses. As in the example above, uncharacterized germplasm has limited value, but its value increases after it has been collected and its attributes are known. Turfgrass breeders attempt to improve multiple traits and attributes including turfgrass quality, genetic color, grow-in and recovery speed, density, texture, disease resistance, drought and cold tolerance, and others. Plant breeders start with a raw product, plant germplasm, and spend multiple years, in most cases 10+ years, on selections of genetic improvement to release products such as cultivars that are of increased value. Revenue from new cultivars must exceed the cost development. Although great efforts and resources are devoted to this process, breeding programs risk little return from investment. Among the many factors that influence success of turfgrass breeding programs, one of the most limiting is the unknown nature of genetics within a breeder's germplasm. Genomics-enabled breeding is understanding and employing that genetic capability. This process leverages information from the modern genomic tools that greatly accelerate breeding cycles to increase genetic gains. Plant genomics aid to characterize the genetic compositions, variations, structures, functions, and networks in plant genomes. By gaining insights into the rich genetic variations in a germplasm panel, breeders may explore the genetic mechanisms of many agronomic traits. At Mississippi State University, one of our turfgrass breeding projects focuses on exploring genomic diversity in the Bermuda grass germplasm using next-generation sequencing technologies. Next-generation sequencing, NGS, is a sequencing technology that offers the ability to generate large amounts of data very quickly. In addition to its application in plant breeding and genetic studies, other notable uses of NGS include clinical diagnostics, such as the identification of new coronavirus strains and other pathogens. In our turfgrass breeding program, we assembled 206 Bermuda grass accessions of worldwide origins and sequenced them using this technology. Over 34,000 molecular variant sites have been detected. This enormous data set will provide unprecedented insights into the genetic variation, phylogeny, and population structure in this important turfgrass species. See Figure 1. Through it, we will gain better understanding of genetic mechanisms of traits of interest through additional linkage and association analyses. The ultimate end goal is to improve breeding efficiency through marker-assisted selection and genomic selection. For all resources associated with this article, check out our show notes. Don't miss an episode. Subscribe on Apple Podcasts, Spotify, or wherever you get your podcasts. You can also visit us at theturfzone.com.
You've been listening to The Turf Zone. For more episodes of The Turf Zone, visit theturfzone.com and subscribe on Apple Podcasts, Stitcher, or your favorite podcast app.